let's go for the next speaker, Peter Zandstra, Director of Michael Smith Laboratories. I'm the Director and Professor of School of Biomedical Engineering at the University of British Columbia. And he will speak about applying system thinking to the development of regenerative medicine. So let's welcome Peter and hold the questions to the very end. Thanks.
Thank you very much for the invitation. It's really fun to be here. I've been asked to speak about a couple of examples from our work that we've applied really different levels of systems thinking, and some quite simplistic, I would say, to translational problems and taken these through to different therapeutics. So just a quick disclosure, some of the work I'm talking about is in a few companies, XLThera and Notch Therapeutics, that I am both a consultant and a co-founder of. So the first example that I'd like to give is with respect to growing blood stem cells as therapies for leukemia. And whenever I think of blood stem cells, it's difficult not to think about the early discoveries by Jim Turrell, Ernest McCulloch, and Lou Simovich, and some of their foundational observations of how stem cells relate to their differentiated progeny, and how a system such as the distributed system, such as the hematopoietic system, could be regulated through feedback control. And I think it's important when we think about systems in systems biology and regenerative medicine, we recognize that the very foundation of stem cell biology was really put in place through observations and mathematical modeling by this physicist and this hematologist, as they started to understand how single cells could form colonies, what the properties of those colonies were, and how the system behavior could interact to explain the responses of those to transplantation and other processes. And one of the early observations was in this figure where they had this dotted line that you see on the bottom with a question: is there feedback control in the system? This motivated us to ask, why is it so difficult to grow blood stem cells when one generates different cell types that are continuously influencing the growth and differentiation of these blood stem cells. And so early on, we developed a very empirical solution to this. Could one match the rate of media addition and dilution of these cultures to the rates of secretion of these inhibitory factors? And indeed, this had a positive effect. But this wasn't really understanding how the complexity in the system, here represented by all these different molecules which are being secreted at different rates, either in static or in so-called fed batch systems. So what we wanted to do, and I think what we want to do in this field, is move from empirical type modeling, where we measure and use pure statistics to try to explain what's going on, to start to integrate some of the underlying molecular structures and mechanisms. I think as we heard in our last speaker, we have to think carefully about how these are integrated into the critical quality attributes and the key measurements that one does during these cultures. And so the idea is, can we move from this more completely empirical way of optimizing these systems to using more and more relevant data into how we define these systems? So taking this one example a little bit further, one can start to say, if there's these secreted factors in the system, can we understand which cells are secreting which factors, what the directionality of these factors are, and what the overall function of the system might be when one modulates the interaction networks through these factors? And this is work that was carried out first by Dan Kerouac and then Wendy Quao. And what they did was use a number of omics technologies to associate different cell types within the hematopoietic system, and the exact names are not important here, with molecules. These molecules are associated with pathways. These pathways are associated with effects. And you had, at some level, a reductionist way of showing this cell communication network. Of course, in any condition, different aspects of this network would change. So what we then did is took this information and started to ask, what are the key properties of the system that one can start to manipulate? And you can think about rebalancing the network according to receptor expression, rebalancing the network according to compartmentalization, whether the cells are in the marrow or in the system, and other aspects of these ways that feedback could be changed by not only the molecular components but the overall niche components within the system. This allowed us to generate bioreactors in which one would take this baseline model information, do a few measurements of key factors within the system, and then control both the growth, the way that media was added, and the types of components that were added to this media in a more automated way. And this is just a picture of that feedback control, where in this case we have an underlying knowledge of the growth curves of different cell types, how it relates to different secreted factors, in this case TGF-beta1, and how that feeds back into the control system of what is driving the bioreactor in this case. And this has had a very significant and positive effect in terms of our ability to grow both total cells and progenitor cells, and data I don't have time to talk about today. We've since implemented the structure behind some of these bioreactor systems in clinical production processes for expansion of blood stem cells, and have treated more than 50 patients to date with expansion systems based on this fed batch control-based perspective. So the second example that I'll give you is in the area of immunotherapies, and I think points to another challenge that the field faces. So when one is generating mature T cells from stem cells, you have a very long and complex differentiation process that you have to go through from those pluripotent stem cells, either in their normal or engineered space, to blood progenitor cells, to progenitor T cells, and eventually to more mature cells. And the approach one can take is one can start to develop technologies to control different phases of these approaches, and there's a lot of complexity with respect to the timing, the dosing, and the qualitative types of molecules which are applied during this differentiation process. And this leads, of course, to different types of outcomes, whether they're progenitor T cells, regulatory T cells, or other types of T cells in the system. And if you step back and you think about just two molecules which are important in those phases, one can think about the notch activation, which is necessary early on during blood induction, but then needs to fall out and fall down towards T cell maturation, and then the TCR-based signaling, which is eventually important for proliferation and maturation of T cells. And the question is, how do we combine the design of the environment to control these systems, for example, to modulate notch and TGR system, when one has a complex multi-step process like this. And so the types of work that we're starting to use to help optimize this are really at three different levels. First are at the more basic level of understanding the mass action kinetics behind the signaling pathways which are activated, and trying to identify key signaling moieties which can be measured along the way. The second one is more the cell population modeling, similar to what we've done in the hematopoietic system, to try to understand what factors either known or that can be discovered through from omic data can be applied to guiding this differentiation process. And then the third, and this came up in Claudia's introduction, is how can we relate, or Anne's introduction as well, how can we relate the key properties of the cells that we measure in vitro to the overall biology of those cells in vivo, ideally in the patient. So this sort of systems pharmacology aspects of cell therapy products is something that's still very poorly understood. Just giving a few examples of this. This is one example where we're trying to understand the relationship between different signaling activation complexes on the surface of a progenitor cell and a differentiation technology. We can estimate how those affect differentiation, activation of the notch reporter gene, and how other molecules, in this case VCAM, might change the interactions of that activation phase. And of course, this is important for us to match the needs of the differentiation system with the signaling timing and processes which are underway. In my final example and data slide, really points out one of the big challenges in the field, and this is data on the signal of the transplanted CAR T in patients as a function of different patient examples. You can see there's many different tracks in which these are eventually reduced within the patient themselves. And it's been very difficult to try to understand what properties of the cells one should measure in order to make predictions around the persistence of T cells and around their effects on tumor. As we start to apply more complex models to these processes, where you have developmental programs around T cells, different T cell populations, naive memory T cells, as well as effector T cells, and then in this case, the tumor mass shown by B, one can start to simulate and extract within these models what are the parameters that have the largest predictive effect on this. And as we get more and more data, we're going to be more and more capable of making these predictions as relevant as possible, which feeds back, of course, into the design and the quality attributes that we can design our cell culture systems around. So what we're showing on this plot is just T cell numbers as a function of different amounts of memory versus naive versus effector cells, and then how that affects the tumor burden within that particular individual case. So I'm going to stop there so we have time for discussion. I think there's really a great opportunity to apply different levels of modeling, and I think one of the discussion points we can have is where does the systems part of mathematical modeling start to become more than just applying simple empirical models, more complex mechanistic models, and eventually the types of wonderful emergent models that this first speaker talked about, where the complexity becomes so large, one has to find ways of extracting key governing principles from those data sets. I think this can be applied through this understand, predict, and eventually control cycle that underlies both discovery and translation, which is important in regenerative medicine, and can help to handle some of the barriers that we're facing, the multi-scale aspects of the system, the dynamics, the heterogeneity, and then the non-linearity, typically due to feedback control and other interactions within these models, eventually, hopefully, to have a big impact on the types of products we produce for patients. So thank you very much.
Great. Thank you, Peter. Very, very interesting. The discussion will be quite challenging in terms of the different layers, and actually looking into the clinical portion of that. How can that be translated to the patients at the end of the day is going to be a challenge, and also definitely the acquisition of the data that we need to talk about a little later.